Rockland Immunochemicals has partnered with the Wistar Research Institute to produce, validate, and distribute a diverse panel of human melanoma cell lines. Trish Brafford, a Wistar research assistant, has worked exclusively with the cell lines and is here to provide tips for culturing the cells to their full potential.
The melanoma cells grow best slightly less than 37, so 36.5 is better. They'll grow at 37, but not temperatures higher than 37. Lower is better. Depending on the melanoma cells, some will die off if the temperature is greater than 37. Some will just slow their growth. You can usually recover them, split them, take the viable cells, put them in a smaller dish, increase the amount of serum. It's possible to recover them. Melanoma cells grow best at 5% CO2, and that has to do with buffering the pH. So the pH of the media should be slightly basic at 7.6. And if you keep your CO2 at 5%, that works the best. So melanoma cells are derived from melanocytes, which produce melanin. So some of the melanoma cells retain this capability, and they produce melanin, which will change the pigment color, or the color of your media, to a brownish color. Some of the melanoma cells, the pigment remains in packages. So if you look under the microscope, you'll see a lot of brown specks among your cells. That's perfectly normal. Our melanoma cells grow best in tumor 2% media. That's what they were derived in. Calcium is very important. So the melanoma cell lines are all very unique. Some of them grow fast, and you can split them 1 to 16, and you'll have a confluent flask in a week. There are other melanoma cells that you want to keep at a high density, or they start to senesce. They would be split maybe a 1 to 2, 1 to 3, and take a good 2 to 3 weeks before they're confluent again. They're all very unique, and there's no general recommendation for all the melanoma cells that would hold. So the melanoma cells, when you're thawing them, each melanoma line has a different viability. I found the more pigmented the cell line is, the worse the viability. They take a while to recover. Most of the cells are dead, so you want to plate those in a small flask to start. Definitely change the media the following day and watch them. If you need to cut back, split them to a smaller flask because you have a lot of death, then definitely do that. Those lines are difficult sometimes to get started. Once they're growing, they grow great, but the thawing process they don't like. So if your cells no longer are responding in an experiment the way they have before, the first thing I would check is mycoplasma. That will alter a cell's response in experiments. The second thing to check is to see if you're working with the same melanoma cell that you thought you were, and that would be DNA fingerprinting. The DNA fingerprint profiles are available online. You can compare to what you receive. The melanoma cell line repertoire that we have has everything in it. We have from matched patients, primary and metastasis. We have from some patients several metastases. Now the clinical data, so we've been making melanoma cell lines for 40 years. So we don't have extensive clinical data on some of these. So we don't know that the metastasis is a secondary recurrence in the same site sometimes, or if it's from another spot on the body. But you can still use those to look at heterogeneity. So in the past, we've had maybe 20 melanoma cell lines that everybody has used. And although we kept making melanoma cell lines, people had their favorites. So this is a huge benefit to the field because these are all new lines now.
To learn more about patient-derived melanoma cell lines, Call 1-800-656-7625 or visit www.rockland-inc.com/melanoma.